Thank you, John, for the introduction. So this is an oligodendrocyte in the cortex, myelinating axons. What I'm going to talk about today is how the propagation delay for neuronally transmitted information is determined by the node of Ranvier in myelinated axons. So I'm going to start out by reviewing myelinated axons and reminding you that conduction speed is a crucial determinant of neural circuit function, and think about how to tune it. I'll then, in the first third of my talk about morphological aspects of node of Ranvier function and how that can be used to tune conduction speed. So I'll look at variability of node and internode length and push the idea that node length is a tunable variable which can adjust conduction speed, and that to do this is particularly energetically favorable compared to putting more myelin on an axon. Then in the last two-thirds of the lecture, I'll turn to the properties of the electrical properties of the nodal membrane and ask the question whether myelinated axons are simply transmission lines of fixed latency. I'll show you that adenosine receptors and IH channels are expressed at sites of action potential generation, show you that adenosine modulates somatic excitability and decreases the conduction speed of myelinated axons. And then I'll look at where adenosine comes from in the brain and show you that uncaging calcium in astrocytes releases ATP, and this release regulates excitability and conduction speed. So let's start with the kind of theoretical problem. Here's the human brain. You need to send information across it from one bit of gray matter through the white matter to another bit of gray matter. And because this takes a long time, about 300 milliseconds for an unmyelinated axon, evolution has come up with the idea of wrapping the axon with lots of membranes of myelin to decrease the capacitance. And this increases the conduction speed by about a factor of 60. And at the same time, this myelin creates these nodes of Ranvier where the voltage-gated sodium channels are located to propagate the action potential. Now, why might we want to tune action potential speed? Well, if you want to send information like this across the brain, it might need to synergize with information coming from another part of the brain, because when there's coincident excitation of neurons, when synaptic currents arrive at the same time, that tends to facilitate the production of more action potentials. And because of the uncertain length of these pathways, you may therefore need a mechanism to tune conduction speed to get the timing right. Because conduction speed, and hence conduction speed, is important for neural circuits to function correctly. And indeed, theoretical work shows that if you mess around with conduction speed, then you mess up neuronal network oscillations, which may be important for cognition. So how is speed tuned? There are various ways you can do this, and this is particularly important if you want to coordinate time-critical physical tasks like juggling or playing the piano. A broadly known way of doing this is to increase the amount of myelin around an axon. This decreases the capacitance, and that will increase the speed. Another way to do it is to play with the internode length. If you increase the internode length, then a larger fraction of the axon is myelinated, so the capacitance is lower and the speed increases. But on the other hand, there's less passive signal transmission between the nodes the longer the internode is, and so that will decrease the speed. There are two opposing effects there. And finally, and much less well known, you can tune the node length. If you increase the node length, that means less of the axon is myelinated, so the capacitance will go up. And in addition, the resistance for current flow from the sodium channels here through the internode will increase, so that will decrease the speed. But if you increase node length by inserting more of the same kind of membrane, you'll be putting more sodium channels in, and that will increase speed. So I'm going to look at these various factors in the first third of my talk. And then in the last two-thirds, I'll look at how tweaking the node electrical properties can adjust conduction speed. So let's start out looking at variability of node and internode length. If you label voltage-gated sodium channels, in this case in the cortex, you can see the length of the node here between the Casper cell adhesion molecules in the paranodes on each side, which glue the myelin onto the axon. And if you look either in the optic nerve or in the cortex, as here, you find that just looking pretty much anywhere, there are lots of different node lengths. Here's a short one, here's a long one in the optic nerve, and the same in the cortex. I've stacked some different length ones up here. And not only is the mean node length different in these two areas, in the optic nerve it's a micron, in the cortex it's about 1.5 microns, but the spread is different as well. So you can see there's a range of optic node lengths here in the optic nerve. It's broader in the cortex. So the node of Ranvier length varies between and within regions, but it doesn't correlate in any way with node diameter. So it's an independent variable from that. However, it does correlate with the number of sodium channels present in the node. So if you integrate the immunohistochemistry signal here over the whole node, you find the signal you get is proportional to node length, rather roughly. And so it's as though the same membrane is inserted with the same density of sodium channels, same number per unit area. And the longer the node, the more sodium channels you've got. And this will be important in a second. Now, what if we look along a single axon? Here's one axon which we fill with a dye, and we can then, this is in the corpus callosum, we can then focus in and look at individual nodes of Ranvier in that axon. And when we do that, we make a remarkable finding, which is along one axon, the node lengths are much more similar than they are between axons. So this is a roughly 1 micron node length axon. This is a roughly 1.5 micron length axon, node length axon. And this is a 2.5 micron node length axon. And another way of looking at that is to plot each of these curves is for one axon. This is the probability distribution in half micron bins of node lengths. Each one is relatively narrow, much narrower than the overall distribution of node lengths in the white matter tract. And that's expressed here in this coefficient of variation. Yet node length varies, has no correlation with internode length. So these seem to be two independently tunable parameters. This raises the question of how axons coordinate their node length at different positions along the axon, and we have no idea how this happens at present. Now, what does it mean? We've investigated this using computer modeling, using a model which is freely available at the Attwell Lab GitHub site, and you can download and run these models from there. And what we're interested in is how the conduction speed will vary as a function of node length if you adjust the node length. Now, this is the observed range of node lengths, in this case in the optic nerve. It's basically the same story for the cortex. So as we increase the node length going through this observed range, what's going to happen to the conduction speed? And I'm going to start out by looking at a situation where we have a constant number of sodium channels in the node, so that we add membrane but not sodium channels to make it longer. When we do that, less of the axon is myelinated, so the capacitance is higher. And in addition, this resistance for current flow along from the sodium channels here into the internode is increased. So both of those factors will decrease the conduction speed. So this dashed curve here for constant channel number just shows a continual decrease. However, I showed you earlier that the number of sodium channels isn't constant. It increases with the node length, implying that you're putting in membrane with the same sodium channel density here to make it longer. And in that case, as you go to shorter node lengths, you've got less sodium channels, and that decreases the conduction velocity down here. The decrease up the top end is the same, but you decrease the number of sodium channels down here, and that means that you get an optimum to this graph of speed against node length, which is somewhere around here. So changes of conduction speed in this, of node length in this observed range are predicted to significantly affect the conduction speed and could be used to tune the arrival time of action potentials. What about the internode length? If you patch clamp oligodendrocytes, the cells that make myelin, and fill them with dye, then you find here at P12, but also in older animals where the internode has finished growing, you find that there are differences in internode length between different brain regions. This is the corpus callosum, this is the cerebellum. And we think this reflects the development of the internode. We've done experiments where we image growing oligodendrocytes in zebrafish, in which the oligodendrocytes express a dye, so we can see the myelin sheaths growing here. And if we have a calcium sensing dye in those sheaths, we can see calcium transients, which occur every so often. And it turns out that the growth of the sheath over a 5-hour period is proportional to the rate of those calcium transients. So the more calcium transients you have, the more the sheath gets longer. Indeed, if you stimulate the axons electrically, which increases the rate of calcium transients, then you get a longer sheath. And if you block action potentials with TTX, you get a shorter sheath. And if you use patch clamping to put a calcium buffer Bacta into the developing sheaths, then you prevent them elongating. So we think the eventual length of the sheath probably reflects the developmental process, which is controlled by calcium transients in the developing oligodendroglia. Now, how does internode length variation affect conduction speed? This is like the graph I showed you earlier for node length, but now I'm plotting speed as a function of internode length. And you can see I've drawn it for 3 different node lengths. They're all broadly the same. All have got an optimum, and there's a maximum speed for each. As you initially increase the internode length, the speed gets faster because more of the axon is myelinated, and so the capacitance is lower. But as the internode length gets longer, eventually less current reaches the next node, and it takes longer to initiate the action potential at the next node. So altering internode length, like altering node length, will also tune conduction speed. And it's interesting that the mean length, typically you find the mean length is longer than the internode length that will give you the maximum speed. And we believe this is an energetic constraint. It turns out this saves energy. If you quantify this as the amount of sodium which comes into the axon on each action potential, you find that for each 1% that you increase the node length, internode length beyond this value which would give you the maximum speed, you reduce energy consumption by 2 or 3%. Now, let's just think briefly about the energetics of tuning conduction speed. I've suggested you could change the number of myelin wraps, or you could change the node length. And it turns out that changing node length is an energetically cool way to do this, because if you work out the amount of membrane you need to add 1 wrap of myelin to a typical corpus callosum axon, that gives you about a 10% speed change. You need to add about 1,000 square microns of membrane to each internode. In contrast, you can produce that 10% speed change by just manipulating 1 square micron of membrane at the node. So changing the node is more energetically efficient and probably also faster to tune the speed of an axon. Okay, I want to turn now to the electrical properties of the nodal membrane and show you that adenosine receptors control those and modulate conduction speed. Now, are white matter conduction delays modulated by chemical agents? We're familiar with all the neurotransmitters and metabolically derived molecules which can modulate synaptic function and neuronal excitability. Does this also happen in the white matter? Is it possible that there are systematic changes in conduction speed? I'm going to try to persuade you that adenosine is a crucial modulator of propagation speed. And we started out by using antibodies to adenosine receptors to look at whether there are sensors of this metabolic derivative in axons. We found no A1 receptors, but we do find A2a receptors in the axon initial segment, for example. There's a soma just here. This is the axon initial segment where the action potential starts. If you label for adenosine 2A receptors, you see there are lots here. They go all the way down to the start of the region where myelination starts. This is antibody to myelin basic protein. And if you plot the density of sodium channels in green and of adenosine receptors in red, you can see there's a good overlap in the axon initial segment. And it's the same at the node of Ranvier. This is the sodium channels bracketed by Caspar labeling at a node of Ranvier. And if you label for adenosine receptors, you see there at the node as well, and their density matches that of sodium channels. Indeed, about 85 or 90% of AISs and nodes express A2A receptors. And interestingly, A2A is a receptor which raises the cyclic AMP concentration within cells. It's GS coupled. And there's an ion channel, IH channels or HCN2 subunits, which are gated by cyclic AMP and open to generate an inward current. And these are classically what form the hyperpolarization activated channels in the heart, for example, and in neurons. And about 50 to 60% of the AISs and nodes express these. And if you measure the length of the AIS, it comes out as pretty similar no matter which of these three markers you use. So can we look at what these adenosine receptors do? We can patch clamp layer 5 pyramidal neurons and puff adenosine. We've put a red dye, Alexa 594, into the cell here, and we then puff adenosine or an analog, CGS, which activates A2A receptors. There's a red dye in this pipette as well, so we can see where it goes. We find that evokes a depolarization of the cell, about 10 millivolts in this case. If you puff ACSF, nothing happens, but adenosine or the A2A agonist both produce this depolarization. Now, what effect is this going to have on the firing of the cell? We're just puffing it on the axon initial segment. If we inject a current into the soma, which evokes a low current, which evokes a steady rate of firing, then this depolarization produced by the adenosine brings the cell closer to threshold, and so you increase the firing rate. Whereas if you're already injecting a high current into the cell and you have a high firing rate, applying adenosine decreases the rate of firing because you get a depolarizing block because you inactivate the sodium channels. So if you plot the firing rate against injected current, there's an increase in firing rate at low currents and a decrease at high currents, and that's just quantified here to show that it's statistically significant. So that's the AIS. What exactly is happening here to produce this depolarization? Well, if you do negative going voltage steps, you find that in these cells, there's a slowly activating inward current, which is this IH, hyperpolarization activated current, mediated by HCN channels. When you puff on adenosine or this agonist, you find this current gets larger. And carrying out a Hodgkin-Huxley kind of analysis using the tails, which are recorded in this little square box here, you can plot the activation curve of the channels. They're activated as you go more negative. The black dots are the control situation. The red dots are with the adenosine present. And you find there's a shift of this curve to the right. It's significant. And that means that more of these channels get activated at the resting potential, for example. And this is produced by cyclic AMP, because if you put cyclic AMP into the neuron from the patch pipette, then you have already got this rightward shift of the activation curve. And when you then apply adenosine, you get no further change. I should have said that these currents are blocked by the IH blocker ZD7288. So at the AIS, adenosine modulates excitability by raising cyclic AMP and increasing IH. It turns out it's the same at the node of Ranvier, and that's really what I'm focusing on now. So Jonathan Lesmie, a very talented postdoc in my lab, developed this preparation where he can patch clamp a layer 5 pyramidal cell, put a dye in the axon to be able to monitor where the nodes are by looking for branch points. Branches only go off where the nodes are. This is in a mouse we got from Diane Sherman and Peter Brophy, where there's GFP in the paranodes, so we can also see the nodes that way. And he then patch clamps the end of the axon where it's been severed by the brain slicing process, and there's a bleb which he can patch. And then afterwards, he can do immunohistochemistry to label for adenosine receptors, shown in blue here, producing this purple color and at the node. So he then waits for a somatic spike to occur and records the resulting spike when it reaches the end of the axon. And then by doing these phase plane plots, we have some arbitrary criteria to define when the start of the action potential is, for example, shown by these dashed lines. And you can see that if you compare these two at the bottom, which are recorded at the bleb here, when you puff the adenosine, there's a significant increase in the delay till when the action potential arrives. So this delay is large and significant, and we can then estimate how that turns into conduction speed by making some assumptions, first of all, about where the action potential is initiated within the AIS, either in the middle or at the end. And we also need to assume something about the forward speed of propagation of the action potential versus the rate at which it goes backwards into the soma. And it turns out the extreme values you get by playing around with all of that are that it's either 2 or 3 times faster going forwards than it is going backwards. And that's these two plots here. No matter what you assume, you get a large change of conduction speed, about 2-fold on this estimate. And I'll validate that with some computations later. So this is a computational model where we use that same model that I showed earlier to simulate a soma with an AIS, then 3 internodes, and then the bleb. And the active membranes are just the AIS and the 3 nodes here. And we can then simulate the action potential spreading through these different compartments. And if we inject, first of all, we assume there's no IH present at all, just have standard Hodgkin-Huxley kind of currents. If we inject half a nanoamp here at the soma to get a nice rate of spiking, if we add IH, implying we're puffing adenosine just at the AIS, then that converts it to a situation where we get a depolarization of 5 millivolts or something. We now only get 1 spike because we've got a depolarizing block. So we're at the high end of that curve I showed you earlier. And in fact, if you plot a graph, putting the adenosine activated IH just into the AIS, then you get this blue curve here. So the more IH you have, the lower you push the rate of firing. If you put it just into the nodes, then you get no effect on the firing rate. And that's because the nodes are relatively electrically isolated from the AIS. So if you look at the resting potential at the soma, as you add IH at the AIS, it gets more and more depolarized, but putting it in the nodes has much less effect. If we look at this graph on the right, you can then do the experiment, the theoretical experiment, the other way around by putting IH just into the nodes. If you put it just into the nodes, you get a profound decrease in conduction speed, whereas if you put it just in the AIS, then you only get a much smaller decrease of conduction speed. And finally, if we simulate an extended axon, not a little stubby axon like this, but if we forget about the soma and the bleb and just have an infinitely long axon going across the corpus callosum, as we put more IH into the nodes, we find we can roughly halve the conduction speed like this. So I've shown you then that adenosine decreases the conduction speed, and the last question to answer then is where might the adenosine come from? Adenosine is classically produced when energy levels get low in the brain, for example, as you're heading towards sleep. Adenosine builds up. I want to show you that the adenosine probably comes from astrocytes. Now, there's a long literature on astrocytes releasing ATP from Stan Cater's work in cultured astrocytes 20 years ago. There's been a lot of work by many people since, including Phil Hayden and his colleagues, which have shown that ATP is released when calcium rises in astrocytes. And that is then converted extracellularly to adenosine. And a recent paper by Anne Schaefer has shown that at least one of the enzymes doing this is present on microglia. So it's a real glial collaboration going on here. And that then can act on synapses to decrease glutamate release or modulate excitability. And in fact, as ATP is released and adenosine builds up as you head towards sleep, this is thought to promote sleep. So this is all in the gray matter. What about myelinated axons in the gray and white matter? If we label for astrocytes by labeling for GFAP, astrocytes, of course, are not just in the gray matter. GFAP actually labels white matter axons better. white matter astrocytes better. And we can then use the preparation that I talked about earlier, where we patch a layer 5 neuron, fill it with dye, and then look afterwards with immunohistochemistry to see where astrocytes are in relation to the path of this axon. And for both of these, for this axon here, if we focus in on particular regions, you can see there's an intimate association of astrocyte processes with the myelinated axon in both of these regions. And this is also true in the white matter itself. Astrocyte processes run in the same direction of the axons. And if you patch clamp oligodendrocytes and fill them with a dye and then label for GFAP, you again find that the oligodendrocyte processes forming the myelin sheaths are quite intimately associated with astrocytes. So does it matter? Well, to look at this, what we've done is to patch clamp astrocytes, in this case with the calcium dye ROD2 inside. And we also have NPEGTA, which is a caged form of calcium. So it's calcium bound to EGTA, which can be released by light. And we detect internal ATP with quinacrine, which labels vesicles of containing ATP. I should say the ROD2 doesn't go into all the small processes of the astrocyte, and it may be these ones which seem to be somewhat detached from the astrocyte process are in finer processes that the ROD2 hasn't gone into. When we uncage astrocyte calcium, and I'll show you a movie of this in a minute, most of these vesicles disappear because they're released into the extracellular space. You can see the number of vesicles is roughly halved. If you do this without the calcium cage in the astrocyte, there's no change in the number. And if you look in adjacent areas away from the astrocyte, there's also no change in these little green blobs. Now, is this ATP being released? We can assess this using luciferin luciferase, which generates a fluorescent light when ATP is present. And here we've just got a dish of this stuff, and we puff some ATP into it to show you get a light out of it, but you don't if you just apply saline. If we then put the cage calcium into the astrocyte and uncage it with our 2-photon laser, you get a big signal due to ATP release. And although there is a bit of an artifact here, you don't get this happening if you don't include the cage calcium in the astrocyte, as you can see from these mean data on the right. So we believe that when calcium goes up in astrocytes, it releases ATP. And this can happen during neuronal activity. Here's an astrocyte around a layer 5 pyramidal neuron. This is its dendrite, this is its axon, this is a blow up of this area. If I play the movie, you'll see that when it says spikes, now there's a gradual rise of calcium both around the dendrite and around the axon. And it turns out that if you just evoke spikes for a second, and they're going at about 30 hertz, then you get a delayed rise in calcium around the dendrites, because of substances released from the dendrites affecting the astrocyte. But if you stimulate for a longer period, then you get a rise around the axon as well as the dendrites. So this establishes that it is possible to get a rise of astrocyte calcium around the axon. And in this case, we now replace the neuronal activity by uncaging again to show that we can raise calcium in these processes here around the axon of this pyramidal cell here. And if we do this whilst recording the membrane potential is depolarized. This is blocked by the IH blocker that I told you about earlier. It's blocked by dialyzing in a blocker of this is the A2a blocker. It's blocked by dialyzing in a blocker of those IH channels, and nothing happens if there's no calcium cage in the astrocyte. So this is just like applying adenosine, and we assume that's what the calcium uncaging is doing. And if you inject current steps into the neuron at the same time, it produces an increase of excitability at low firing rates and a decrease at high firing rates, just like we saw with adenosine. And if you block A2a receptors, that abolishes this effect. And if you block IH channels, that abolishes the effect as well. So we think that uncaging calcium in astrocytes releases adenosine, which has the same effects on the AIS as I showed you for applying adenosine, and it's the same for the node of Ranvier. Again, we do use this stubby axon preparation filled with a dye, and we uncage calcium in an astrocyte here, and later on we can do immunocytochemistry to show that the astrocyte extends around the nodes of Ranvier. And again, we find a decrease of speed of the axon, just like we saw when we were applying adenosine itself. So to sum all of that up, when neuron activity increases, that increases calcium in astrocytes, and this can spread to the processes near the axon. I already showed you that the morphology of the node of Ranvier can be used to change the speed of action potentials going along these axons. But in addition, release of ATP and conversion to adenosine activates A2a receptors, which raise cyclic AMP. They activate an inward current through these IH channels, and that changes the conduction velocity, as I've shown you today. So the change of conduction speed may disrupt information processing by altering synchrony of arrival of action potentials. It may disrupt generation of oscillations, which depend on timed communication between different parts of the brain, and it may contribute to functional disconnection as sleep approaches. Thank you for listening. These are the people who did the work, and I'm happy to take questions.